Hi class, this is your instructor, Skylar Huff, and welcome to Chapter 3, The Chemical Building Blocks of Life. Despite the complexity and diversity of living systems, all organisms share the same basic chemistry. In the last chapter, you all saw how this involves a limited number of elements. Now we will see that organisms consist of a few basic kinds of molecules. These molecules are all built on a carbon framework, making them organic molecules. These are all synthesized by living things and are called macromolecules because they are very large molecules. Like the DNA shown at the right and the protein shown at the left and that bottom right along with that DNA, they are huge. So we recognize 4 classes of biological macromolecules. So as you see before yourselves, they are carbohydrates at 3.2. From there we'll go to the lipids in 3.5. We'll go right up to proteins in 3.4 and end things with nucleic acids in section 3.3. And that is the order that we will follow. So 3.2, 3.5, 3.4, and then 3.3. So let us begin with a basic introduction by way of carbon. So in Chapter 2, we reviewed the basics of anatomic structure and chemical bonding. Biological systems obey all the laws of chemistry. Thus, chemistry forms the basis of living things. The framework of biological molecules consists of predominantly carbon atoms bonded to other carbon atoms or to atoms of hydrogen, oxygen, nitrogen, sulfur, or phosphorus. And because carbon atoms can form up to 4 covalent bonds, molecules containing carbon can form straight chain branches or even rings, balls, tubes, and coils. Molecules consisting of only carbon and hydrogen are called hydrocarbons. So because the oxidation of hydrocarbon compounds results in a net release of energy, hydrocarbons make good fuels. Gasoline, for example, is rich in hydrocarbons. And propane gas, another hydrocarbon, consists of chains of 3 carbon atoms with 8 hydrogen atoms bound to it. And given that, that is why. So theoretically speaking, the length of chain of carbon atoms is unlimited. As described in the rest of this chapter, the 4 main types of biological molecules often consist of huge chains of carbon-containing compounds. So now, carbon and hydrogen atoms both have very similar electronegativities. Electrons in that carbon-carbon or that carbon-hydrogen bonds are therefore evenly distributed, with no significant differences in charge over the surface, or at least over the molecular surface. For this reason, hydrocarbons are non-polar. Most biological molecules produced by cells, however, contain other atoms because these other atoms frequently have different electronegativities. And you can see in your textbook, table 2.2 showing you that. Molecules containing them exhibit regions of partial positive or negative charge. They are polar. These molecules can be thought of as a carbon-hydrogen core to which specific molecular groups called function groups are attached. One such common function group is OH, and that is called the hydroxyl group. You can see the hydroxyl group shown right there at the top, and this is figure 3.2. Functional groups have definite chemical properties that they retain no matter where they occur. Both the hydroxyl and the carbonyl groups, for example, are polar because of the electronegativity of oxygen atoms. Other common functional groups are the acidic carboxyl, the phosphate group, and the basic amine group or amino group, NH2. Many of these functional groups can also participate in hydrogen bonding. Hydrogen bond donors and acceptors can be predicted based on their electronegativities shown in Table 2.2. So shown here in Figure 3.2, it illustrates these biologically important functional groups and lists the macromolecules in which they are found. So keep in mind that you'll have hydroxyl groups in carbohydrates. You have those in lipids. Those will be in proteins and nucleic acids. Carbonyl groups will be in the carbohydrates and nucleic acids. Carboxyl groups will be in proteins and lipids. The amine group or amino group will be in proteins and nucleic acids. The sulfhydryl group is in proteins. Phosphate groups are found in nucleic acids. That's their phosphate backbone, so to speak. And then the methyl group will be found in proteins and nucleic acids. Isomers. So we have organic molecules that have the same molecular or empirical formula can exist in different forms called isomers. If there are differences in the actual structure of their carbon skeleton, we call them structural isomers. In our next section, you will see that glucose and fructose are structural isomers, C6H12O6. Another form of isomers, called stereoisomers, have the same carbon skeleton but differ in how the groups attached to the skeleton are arranged in space. Enzymes in biological systems usually recognize only a single specific stereoisomer. A subcategory of stereoisomer are called enantiomers, and are actually mirror images of another. A molecule that has a mirror image version is called a chiral molecule, and when carbon is found in 4 different molecules, this inherent asymmetry exists. And you can see that right here in Figure 3.3. So these are chiral molecules. So when carbon is bound to 4 different groups, the resulting molecule is said to be chiral, such as your hand. So if you see your hand look this way, if you flip your hand right that way, you have that mirror image. So the 2 molecules shown have the same 4 groups, but cannot be superimposed, much like your 2 hands, which cannot be superimposed, but must be flipped to match. So let's get into macromolecules. Remember that biological macromolecules are traditionally grouped into carbohydrates, lipids, proteins, and then nucleic acids. See Table 3.1 as shown to you here. In many cases, these macromolecules are polymers. A polymer is a long molecule built by linking together a large number of small, similar chemical subunits called monomers. They are like railroad cars coupled to form a train. Or you could say they're like Legos. I mean, the Legos on the floor are just Legos on the floor. However, when you build something, that would be that macromolecule built of those small subunits called monomers to make whatever it is that you have built. The nature of a polymer is determined by its constituent monomers. For example, the complex carbohydrate starch is a polymer composed of subunits of the simple sugar glucose. Nucleic acids, DNA, and RNA are polymers of nucleotides, and proteins are polymers of amino acids. And you can see that in Figure 3.4. Now being shown to you all is Figure 3.4. So what you see at the shown here, it doesn't show a protein for you. Nonetheless, these long chains are built via chemical reactions termed dehydration reactions, or you may have heard of them otherwise called a dehydration synthesis reaction. And they are broken down by hydrolysis reactions. Lipids are macromolecules, but they really don't follow the monomer-polymer relationship, or the way we've taught that along the lines of the carbohydrates, the proteins, and nucleic acids. However, lipids are formed through dehydration reactions, which link the fatty acids to glycerol. So here, proteins are shown in figure 3.4, and you see the intermediate filament, you see the polypeptide there in the center, and the amino acid there at that right-hand side. And then at the bottom, you find there the lipid. And as mentioned moments ago, they don't follow that monomer-polymer relationship. Despite the differences between monomers of these major polymers, the basic chemistry of their synthesis is similar. To form a covalent bond between two monomers, an OH group, so to form that simple covalent, or that basic covalent bond, what you have is that hydroxyl group is removed from one monomer, and then the hydrogen atom is removed from the other. And that is shown to you here in figure 3.5a. This reaction is the same for joining nucleotides when synthesizing DNA, or joining glucose units together to make starch. This reaction is also used to link fatty acids to glycerol and lipids. This chemical reaction is also called condensation. So it could be a condensation reaction, or it could be called a dehydration reaction. And this is because of the removal of that hydroxyl as well as that hydrogen atom. And this is the same as the removal of water, hence the name condensation or dehydration reaction. Because for every subunit added to a macromolecule, one water molecule is removed. These and other biochemical reactions require that the reacting substances are held together and that they are in the correct chemical bonds and are stressed and broken. So again, I say, these and other biochemical reactions require that the reacting substances are held close together and that the correct chemical bonds are stressed and broken. This process of positioning and stressing is termed catalysts, and is carried out within cells by enzymes. So there you have it. Enzymes catalyze reactions. The hydrolysis reaction. So cells disassemble polymers into their constituent monomers by reversing the dehydration reaction. A molecule of water is added instead of removed. So again, hydrolysis, I guess by way of its name, is to split, i.e. lysis, with water, hydro. So you can see this in figure 3.5b at that bottom right, to split with water. In this reaction, called hydrolysis, a hydrogen atom is attached to one subunit and a hydroxyl group to the other, breaking the covalent bond, joining the subunits. When you eat a potato, which contains starch, your body breaks the starch down into glucose units by hydrolysis. The potato plant built the starch molecules originally by dehydration reactions. Now on to our first of 4 types of macromolecules, the carbohydrates. Carbohydrates are a loosely defined group of molecules that all contain carbon, hydrogen, and oxygen in a molar ratio of 1 to 2 to 1. Their empirical formula, which lists the number of atoms in the molecule by way of subscripts, shown here, is CH2ON, where N is the number of carbon atoms. Because they contain many carbon-hydrogen bonds, which release energy when they are rearranged, carbohydrates are well suited for energy storage. Sugars are among the most important energy storage molecules, and they exist in several different forms. The simplest form of carbohydrates are the monosaccharides. So mono, single, mono, one. Simple sugars are as few as 3 carbon atoms. But those that play the central role in energy storage have 6. And make sure you look at figure 3.6. And I'll show that for you all here now. The empirical formula of 6-carbon sugar is C6H12O6. And I will not give that formula. You see there in parentheses. I will not use that. I'll just use C6H12O6, such as for glucose. 6-carbon sugars can exist in a straight chain form, but dissolve in water, that aqueous environment. They almost always form rings. So you see here in this order, it says glyceraldehyde at 3-carbon sugar. You have those 2 5-carbon sugars. You all should know these. Ribose. And then there is deoxyribose. And then you have those 6-carbon sugars. You all should know these as well. They are glucose, fructose, and galactose. Make sure you all learn these. These are examples of monosaccharides, the 5-carbon sugars, and those 6-carbon sugars. The most important 6-carbon sugar used for energy storage is glucose, which appeared in the photosynthesis reactions in Chapter 2. Glucose has 7 energy-storing carbon-hydrogen bonds, shown to you all now in figure 3.6, though it is also shown again in figure 3.7, which is right here before you. Depending on the orientation of the carbonyl group, when the ring is closed, glucose can exist in 2 different forms, alpha glucose or beta glucose. This is significant, and we will see when glucose is used as a monomer to form polymers. Glucose is not the only sugar with the formula C6H12O6. Both structural isomers and stereoisomers of this simple 6-carbon skeleton exist in nature. Fructose is a structural isomer that differs in the position of the carbonyl carbon. Galactose is a stereoisomer that differs in the position of the hydroxyl and the hydrogen groups relative to the ring in figure 3.8. These differences often account for substantial functional difference between isomers. Your taste buds can discern them too. Fructose tastes much sweeter than glucose, despite the fact that both sugars have identical chemical composition. Enzymes that act on different sugars can distinguish both the structural and stereoisomers of this basic 6-carbon sugar, or this 6-carbon skeleton. The different stereoisomers of glucose are also important in the polymers that can be made using glucose as a monomer, as you will see later in this section. Most organisms transport sugars within their bodies. In humans, you and me, the glucose that circulates in the blood does so as a simple monosaccharide. In plants and many other organisms, however, glucose is converted into transport form before it is moved from place to place within the organism. In such form, it is less readily metabolized during transport. Transport forms of sugars are commonly made by linking 2 monosaccharides together to form a disaccharide. Di means 2. Disaccharides serve as effective reservoirs of glucose because the enzymes that normally use glucose in the organism cannot break the bond linking the 2 monosaccharide subunits. Enzymes that can do so are typically present only in the tissue that uses glucose. Let us continue. So shown before you are examples of the monosaccharide here, and then from such what makes up said monosaccharide. In other words, sucrose, the disaccharide, is made up of alpha glucose and fructose. So by way of that dehydration reaction or that condensation reaction, we get sucrose. And maltose is also shown there. Other examples include lactose So we have sucrose, lactose, and maltose, used for sugar transport and energy storage. You all should know this. So transport forms differ depending on which monosaccharides are linked to form the disaccharide. Glucose forms transport disaccharides with itself and with many other monosaccharides, including fructose and galactose. When glucose forms a disaccharide with the structural isomer fructose, the resulting disaccharide is sucrose, as I just mentioned. That is regular old table sugar. That's shown in figure 3.9a. Sucrose is the form most plants use to transport glucose and is the sugar that most humans and other animals eat. Sugarcane and sugar beets are rich in sucrose. When glucose is linked to the stereoisomer galactose, the resulting disaccharide is lactose, or milk sugar. Milk sugar. Many mammals supply energy to their young in the form of lactose. Adults often have greatly reduced levels of lactase, the enzyme required to cleave lactose into its two monosaccharide components, being glucose and galactose. Thus, they cannot metabolize lactose efficiently, and it's all due to the adults having a greatly reduced level of lactase. So lactase is that enzyme that breaks down lactose. So this can result in lactose intolerance, which you may have heard of. Most of the energy that is channeled into lactose production is therefore reserved for offspring. For this reason, lactose is an energy source primarily for the offspring in mammals. Let's continue on. Polysaccharides are long polymers made up of monosaccharides that have been joined through dehydration reactions. Starch, a storage polysaccharide, consists of entirely alpha glucose molecules linked in long chains, whereas cellulose, a structural polysaccharide, also consists of glucose molecules linked in chains, but these molecules are beta-glucose. That's that major difference between starch and cellulose. Because starch is built from alpha-glucose, we call the linkages alpha linkages, whereas the cellulose has beta linkages. So this also gets to the digestion of such. We cannot digest cellulose, though we can digest starch. Starches and glycogen. Organisms store metabolic energy contained in monosaccharides by converting them into disaccharides, such as maltose, shown in figure 3.9b. 3.9b. That is not shown here. These are then linked together into the insoluble polysaccharide called starches. These polysaccharides differ mainly in how the polymers branch. The starch is the simplest structure. Well, the starch with the simplest structure is called amylose or amylose. So amylose, it is composed of many hundreds of alpha glucose molecules linked together in long unbranched chains. Each linkage occurs between the carbon 1 or C1 of glucose molecule and the C4 of another, making them alpha 1 to 4 linkages, shown in figure 310a. The long chains of amylose tend to coil up in water, a property that renders amylose insoluble. So potato starch is about 20% amylose. Most plant starch, including the remaining 80% of potato starch, is somewhat more complicated variant of amylose called amylopectin. Pectins are branched polysaccharides, with the branches occurring due to bonds between the C1 of one molecule and the C6 of another, so those alpha C1 to C6 linkages. These short amylose branches consist of 20 to 30 glucose subunits. The comparable molecule to starch in animals is glycogen. Yes, and I won't say it, but I guess I'll say it. I guess I'll say glycogen is starch for animals. But yes, it's the comparable starch in animals is glycogen. Like amylopectin, glycogen is an insoluble polysaccharide containing branched amylose chains. Glycogen has a much longer average chain length and many more branches than plant starch. So there you have it. We have starch by way of figure 310b, and we have glycogen by way of figure 310c. Into cellulose. Although some chains of sugars store energy, others serve as structural material for cells. For two glucose molecules to link together, the glucose subunits must be of the same form. Cellulose is a polymer of beta-glucose, and you can see that here in figure 311. Figure 311. The bonds between adjacent glucose molecules still exist between the C1 of the first glucose and C4 of the next glucose, but these are beta linkages, beta 1 to 4 linkages. The properties of a chain of glucose molecules consisting of all beta-glucose are very different from those of starch. These long, unbranched beta-linked chains make tough fibers. Cellulose is the chief component of plant cell walls. It is chemically similar to amylose, with one important difference. The starch hydrolyzing enzymes that occur in most organisms cannot break the bond between two beta-glucose units because they only recognize alpha linkages. Because cellulose cannot be broken down readily by most animals, it works well as a biological structural material. But some animals, such as cows, are able to utilize cellulose aided by symbiotic bacteria and protists in their digestive tract. These organisms provide the necessary enzyme to cleave that beta 1 to 4 linkage, releasing glucose further for metabolism. And it's all because of their gut bacteria and gut protists. And now let's get on to what is called chitin, which is not shown here. What chitin is, however, shown to the far right corner. Chitin, the structural material found in arthropods and many fungi, is a polymer of N-acetylglucosamine and a nitrogen-containing derivative of glucose. When cross-linked by proteins, it forms a tough, resistant surface material that serves as the hard exoskeleton of insects and crustaceans. Few organisms are able to digest chitin. I repeat, few organisms are able to digest chitin, but most possess chitinase, the enzyme, and it probably to protect against fungi. So this is and has been the carbohydrates. Lipids. Lipids are a somewhat loosely defined group of molecules. But the main chemical characteristic, they are insoluble in water. I repeat, lipids are insoluble in water. Storage fats such as animal fat are one kind of lipid. Oils such as those from olives, hint hint, extra virgin olive oil, corn, and coconut are also lipids, as are waxes such as beeswax and even the wax in your ear right now. Even some vitamins are lipids. Lipids have a very high proportion of nonpolar carbon-hydrogen bonds, and so long chains of lipids cannot fold up like a protein to confine their nonpolar portions away from their surrounding aqueous environment. Instead, when they are placed in water, many lipid molecules spontaneously cluster together and expose what polar hydrophilic groups they have to the surrounding water, while confining their nonpolar hydrophobic parts of the molecules together within the cluster. You may have noticed this effect when you add a pan containing water to oil. So if you add oil in a pan of water, the oil beads up into cohesive drops on the water's surface. This spontaneous assembly of lipids is of paramount importance to cells, as it underlies the structure of cell membranes. Many lipids are built from a simple skeleton made up of two main kinds of molecules, fatty acids and glycerol. Fatty acids are long-chain hydrocarbons with a carboxyl group at one end, and then the glycerol is a 3-carbon polyalcohol So many lipid molecules consist of a glycerol molecule with 3 fatty acids attached, 1 to each carbon of the glycerol backbone. Because it contains 3 fatty acids, a fat molecule is commonly called a triglyceride. The more accurate chemical name is a triglycerol. So triacylglycerol. This basic structure is depicted in figure 3.28, shown to you all now. The 3 fatty acids of a triglyceride need not be identical, and often they are very different from one another. The hydrocarbon chains of the fatty acids vary in length. The most common are even-numbered chains of 14 to 20 carbons. The many carbon-hydrogen bonds of fat serve as a form of long-term energy storage. I repeat, long-term energy storage. If all of the internal carbon atoms in a fatty acid chain are bonded to 2 hydrogen atoms, we call this saturated, with the maximum number of hydrogen atoms possible. And you'll see this in figure 3.28 as I just showed. A fatty acid with double bonds between one or more pairs of successive carbon atoms will have fewer hydrogen atoms and thus is unsaturated. So fatty acids with one double bond are monounsaturated, and those with more than one double bond are termed polyunsaturated. Most naturally occurring unsaturated fatty acids have double bonds with a cis configuration where the carbon chain is on the same side before and after the double bond. And you can see this in figure 3.28b. And you can see that there are double bonds here. So double bonds. So in case you need help, at B on the right hand side, that is the unsaturated fat. And as you can see right there, they have one or more double bonds. So double bond here and there, double bond there, and then 3 more double bonds here. Whereas here at A, we have what is called a saturated fatty acid. They're all in single bonds. So if the carbon chain is on the opposite side before and after the double bond, it is a trans configuration. So the presence of that double bond in fatty acids affect the melting point of that triglyceride because they lack the free rotation about the cis double bonds and produce a kink in the chain. This makes unsaturated fats liquid at room temperature. I repeat, liquid at room temperature are unsaturated fats. They're referred to as oils, while most saturated fats, such as the animal fat and those in butter, are solid at room temperature. You all should know this. You need to know this if you did not know this before now. For many years, the consumption of saturated animal fats was linked to increased risk of cardiovascular disease, and because of this, the food industry started producing solid products such as margarine by adding hydrogen to unsaturated fats to make them partially saturated. This process, called partial hydrogenation, was intended to produce a healthier product. They did not know what they were doing. Unfortunately, the process also converts cis double bonds to trans double bonds, creating so-called trans fats. Those are horrible for your and my health. And it's because they are less kinked. They are solid at room temperature, but they have also been linked to elevated levels of low-density lipoprotein, i.e. your bad cholesterol, and lowered levels of high-density lipoprotein, HDL, meaning your good cholesterol. You should know about those two, the ratio between your HDL, which is that good cholesterol, and the HDL, the high-density lipoproteins, are higher in protein, whereas the LDL, called the low-density lipoproteins, are higher in fat content. So the risk for cardiovascular disease from trans fats appears to be greater than that due to the saturated fat. The attempt to produce something more healthy actually produces something less healthy. That's in your textbook, you all. So trans fats have been removed from the FDA's list of generally regarded as safe compounds, which has helped to remove trans fats from common sources. So placed in water, triglycerides spontaneously associate together, forming fat globules that can be very large relative to the size of the individual molecules. Because fats are insoluble in water, they can be deposited at specific locations within an organism, such as in vesicles of adipose tissue. So organisms that contain many other kinds of lipids besides fats, such as the terpenes, which are long-chain lipids that are components of many biologically important pigments, such as chlorophyll, and the visual pigment retinol, are composed of 4 carbon rings. 4 carbon rings. Most animal cell membranes contain the steroid cholesterol. Other steroids, such as testosterone and estrogen, function as hormones in multicellular animals. Prostaglandins are a group of about 20 lipids that are modified fatty acids with 2 nonpolar tails attached to a 5-carbon ring. Prostaglandins act as local chemical messengers in many vertebrate tissues, though that won't be taught in this class. Fats are excellent energy storage molecules. I repeat, they are excellent energy storage molecules. Most fats contain over 40 carbon atoms, and the ratio of carbon-hydrogen bonds in fats is more than twice that of the carbohydrates. This means that fats are relatively more reduced than carbohydrates and will thus release more energy upon oxidation. This makes fats much more efficient molecules in the storage of chemical energy. So on average, fats yield about 9 kilocalories of chemical energy per gram, as compared with about 4 kilocalories of energy per gram for carbohydrates. So more than 2 times that amount. Most fats produced by animals are saturated except some fish oils, which is why fishes are typically recommended as a dietary food to eat. More healthy than, let's just say, a steak. The exceptions are the tropical plant oils. I repeat, the exceptions are the tropical plant oils. If you say, what do I mean? I here refer to palm oil, likely found in peanut butter that you eat, and coconut oil, likely now shown on, let's just say, social media by some influencer as being healthy. It is not. And I say that because these are saturated even though they are liquid at room temperature. It's palm oil that makes peanut butter creamy. So if the peanut butter has a separation of oil there, meaning there's lots and lots of oil at the top or maybe at the bottom, That's because it has no palm oil, and you have to stir it. So when an organism consumes excess carbohydrate, it is then converted into starch, glycogen, or fats reserved for future use. The reason that many humans in developed countries gain weight, the reason that many people in developed countries gain weight as they grow older is that the amount of energy they need decreases with age, yet their intake of food does not. So instead of asking for a GLP-1, maybe think back to the consumption of calories. Thus, an increasing proportion of carbohydrates they ingest is converted into fat. And this is in your text. A diet heavy in fats is one of several factors thought to contribute to heart disease, particularly atherosclerosis. In atherosclerosis, sometimes referred to hardening of arteries, and it's referred to that, but it's actually that lipid plaque buildup. The fatty substances called plaque, they adhere to the lining of blood vessels, so that is what atherosclerosis is. So that occludes the lumen of the artery, blocking the flow of blood. And I'll be more specific and say it's called arteriosclerosis, that is that hardening of the arteries. Whereas atherosclerosis is that lipid plaque buildup. You all should know this, class. That's why I'm teaching this. And then fragments of plaque can break off from a deposit and clog arteries to the brain, which would then cause a stroke. Take care of yourself out there, class. The phospholipid. So complex lipids called phospholipids are among the most important molecules of the cell because they form the core of all biological membranes. An individual phospholipid can be thought of as a substituted triglyceride. That is, a triglyceride with a phosphate replacing one of those fatty acids. The basic structure of the phospholipid includes 3 subunits. We have the glycerol head region. We have those fatty acid tails. There are 2 of those, and that phosphate group. So there you have it, glycerol, fatty acid, and the phosphate. And this forms all biological membranes. So the phospholipid molecule can be thought of as having a polar head at one end and 2 long, very nonpolar tails at the other end. So if you hear me saying it, I'll be saying a glycerol or a glycerol head that is polar, and those 2 nonpolar fatty acid tails makes up the phospholipid. And that's shown to you here. So this structure is essential to how these molecules function, although it first appears paradoxical. Why would a molecule need to be soluble in water, but also not soluble in water? So it's the formation of the membrane that shows the unique properties of a structure. So in water, the nonpolar tails of a nearby lipid molecule aggregate away from water, forming spherical micelles with the tails facing inward, which you're seeing now, tails facing inward. This is actually how detergent molecules work to make grease soluble in water. The grease is soluble within the nonpolar interior of the micelle, and the polar surface of the micelle is soluble in water. With phospholipids, the more complex structure forms in which 2 layers of the molecule line up with the hydrophobic tails of each layer pointing toward one another, or a inward leaving the hydrophilic heads orienting outward, as you can see here. So fatty acid tails inward, glycerol heads pointing outward that are hydrophilic. Hydrophilic tails inward. So this is forming that bilayer, or as you hear me call it, that lipid bilayer, or that phospholipid bilayer. You all should know this. So lipid bilayers are the basic framework of biological membranes, and we'll get to this in chapter 5. But this is why it's said to be an amphipathic phospholipid bilayer. So that has been the lipids. Now let's get on into what are called proteins. So proteins are the most diverse group of biological molecules, both chemically and functionally. Because proteins have so many different functions in cells, we cannot begin to list them all. Yeah, this is not all of those. But you list them anyway, publisher. Thanks a lot. Nonetheless, we can however group these functions into the following 7 categories. This list is a summary only. However, the function of the proteins, it is relevant to most topics in biology. And if you say, well, whatever do you mean? What I mean is this. Enzymes can function in enzyme catalysts. So enzymes are biological catalysts that facilitate specific chemical reactions. Because of this property, the appearance of enzymes was one of the most important events in the evolution of life. Most enzymes are 3-dimensional globular proteins that fit snugly around molecules they act on. This fit facilitates chemical reactions by stressing particular chemical bonds. Next up is defense. Other globular proteins use their shapes to recognize foreign microbes and cancer cells. These cell surface receptors form the core of the body's endocrine and immune systems. They keep you alive. Transport. A variety of globular proteins transport small molecules and ions. The transport protein, hemoglobin for example, transports oxygen in the blood. Membrane transport proteins help move ions and molecules across a membrane, such as, let's just say, chemically gated, or some ligand or ligand gated, or even an ion. So gated channels can be gated even by membrane potential. So they are gated by electricity. Next up is support. Support. Protein fibers play structural roles. These fibers include keratin, such as in your hair, fibrin, such as in blood clots, and even collagen. The last one, collagen, forms the matrix of skin, ligaments, tendons, and bones, and is the most abundant protein in a vertebrate body. Next up is motion. Muscles contract through sliding motion of 2 kinds of protein filaments, known as actin and myosin. And those contractile proteins also play a key role in the cell's cytoskeleton and in moving materials within cells. Regulation. Small proteins called hormones, yes, hormones, serve as intercellular messengers in animals. Proteins also play many regulatory roles within the cell, turning on and shutting off genes during development, for example. And in addition, proteins receive information acting as cell surface receptors. And lastly, storage. Calcium and iron, that's iron, Fe, are stored in the body by binding as ions to storage proteins. So if you look at table 3.232, it lists protein functions, classes of proteins, examples of each of those, and then examples of use. And throughout this class, we will get to some of these, a number of these. All of these, never. Some of these, surely. Use that, please. So proteins, what are they? Proteins are linear polymers made with only 20 different amino acids. As their name suggests, they contain an amino group, NH2, an acidic carboxyl group, and there you have it. So the specific order of amino acids determines the protein's structure and function. So many scientists believe amino acids were among the first molecules formed on Earth. It seems likely that the oceans that existed early in the history of the Earth contained a wide variety of amino acids. The generalized structure of an amino acid, as shown, of the amino and carboxyl groups bonded to a central carbon atom, with an additional hydrogen and functional side group indicated by R. So that's the R group, or what has been referred to as an R group. These components completely fill the bonds of that central carbon. The unique character of each amino acid is determined by the nature of the R group. So notice, unless the R group is an hydrogen atom, as in glycine amino acids are chiral and can exist in two anti-enteric forms, D or L. So in living systems, only the L amino acids are found in proteins, and the D amino acids, they are rare. The R group also determines the chemistry of amino acids. Serine, in which the R group is CH2OH, is a polar molecule. Alanine, which has CH3 as its R group, is nonpolar. So the 20 common amino acids are grouped into 5 chemical classes based on their R group. So there are the ones which are non-polar amino acids, such as leucine. There are the polar uncharged amino acids, such as threonine. There are the charged amino acids, such as glutamic acid. There are aromatic amino acids, such as phenylalanine. And then there are amino acids that have special functions and unique properties. So examples are methionine, which is often that first amino acid in the chain of amino acids. And then you have proline, which causes kinks in chains, and even cysteine. So each amino acid affects the shape of a protein differently, depending on the chemical nature of its side group. For example, portions of a protein chain with numerous nonpolar amino acids tend to fold into the interior of the protein by hydrophobic exclusion. So there you have it. These are the amino acids and amino acid structure. Peptide bonds. In addition to its R group, each amino acid is ionized at the physiological pH with a positive amino group at one end and a negative carboxyl group at the other end. The amino and carboxyl groups on a pair of amino acids can undergo dehydration reaction to form a covalent bond, shown here. This is figure 3.20. The covalent bond that links two amino acids is called a peptide bond. The two amino acids linked by such a bond are not free to rotate around the N and C linkage because the peptide bond has a partial double bond character. This is different from the NC and the CC bonds central to the amino acid. This lax rotation about the peptide bond is one factor that determines the structural character of the coils and regular shapes formed by amino acids. An unbranched chain of amino acids linked by peptide bonds is called a polypeptide. If a polypeptide can fold into a structure with some function, such as the antimicrobial enzyme lysozyme in your saliva, we call it a protein. However, some proteins, such as the oxygen-carrying protein hemoglobin in your blood, consists of more than one polypeptide. So the pioneering work of Frederick Sanger in the early 1950s provide the evidence for each kind of protein has specified amino acid sequence. Using chemical methods such as removing successive amino acids, then identify them. Sanger succeeded in determining the amino acid sequence of insulin. In doing so, he demonstrated clearly that this protein had a defined sequence, which was the same for all insulin molecules in the solution. Although many different amino acids occur in nature, only 20 commonly occur in proteins. Of these 20, 8 are called essential amino acids because humans cannot synthesize them, thus must get them in their diets. So figure 3.21 illustrates these 20 amino acids and their side groups. It is shown to you all now. So now let's get to levels of protein structure and finish up this chapter. There are 4 levels of protein structure, and the shape of protein determines its function. One way to study the shape of something as small as a protein is to look at it with very short wavelength energy. In other words, X-rays. X-rays can be passed through a crystal of protein to produce a diffraction pattern. This pattern can then be analyzed, and I say as it's analyzed, by a painstaking procedure that allows the investigator to build up a 3-dimensional picture of the position of each atom. The first protein to be analyzed in this way was myoglobin, and the related protein hemoglobin was analyzed soon thereafter. As more and more proteins were studied, the general principle became evident. In every protein studied, essentially all of the internal amino acids are nonpolar ones. Amino acids such as leucine, valine, and phenylalanine. Water's tendency to hydrophobically exclude nonpolar molecules literally shoves nonpolar portions of the amino acid chain into the protein's interior. This tendency forces the nonpolar amino acids into the close contact with one another, leaving little empty space inside. Polar and charged amino acids are restricted to the surface of the protein, except for the few that play key functional roles. So the structure of proteins is usually discussed in terms of hierarchy of 4 levels: primary, secondary, tertiary, and quaternary, shown in figure 3.22. The primary structure of a protein is its amino acid sequence because R groups that distinguish the amino acids play no role in the peptide backbone. So a protein can consist of any sequence of amino acids, thus any 20 different amino acids may appear at any position. A protein containing 100 amino acids could form from any 20 to the 100th power different amino acid sequences. That's the same as 10 to the 130th, or 1 followed by 130 zeros, more than the number of atoms known in the universe. This important property of proteins permits great diversity. Please remember that. So consider the protein hemoglobin, the protein in your blood. It uses to transport oxygen. The protein in your blood uses to transport oxygen. Hemoglobin is composed of 2 alpha-globin peptide chains and 2 beta-globin peptide chains. The alpha-globin chains differ from the beta-globin ones in the sequence of amino acids. Furthermore, any alteration in the normal sequence of either types of globin proteins, even by a single amino acid, can have, and I guess I'll say it can have drastic effects on how the protein functions. The secondary structure. The amino acid side groups are not only portions of proteins that form hydrogen bonds. The polypeptide backbone consists of repeating units of amino acids, each with an amino acid and a carboxyl group that can participate in hydrogen bonds. These hydrogen bonds can be with water or other amino acids in the backbone. If too many hydrogen bonds formed with water, the protein would tend to behave like a random coil and wouldn't produce the kinds of globular structures that are common in proteins. Linus Paulding suggested that amino and carboxyl groups could interact with one another if the peptide was coiled into a spiral that he called the alpha helix. We now call this sort of interaction with groups in the peptide backbone the secondary structure. Another form of secondary structure can occur between regions of peptide aligned next to each other to form a planar structure called the beta sheet, and you may have heard of it as a beta pleated sheet. These can be either parallel or anti-parallel depending on whether the adjacent sections of the peptide are oriented in the same direction or the opposite direction. These two kinds of secondary structure creates regions of proteins that are cylindrical or alpha helices and planar called beta sheets. A protein's final structure can be included in regions of each type of secondary structure. For example, DNA binding proteins usually have regions of alpha helix that can lie across DNA and interact directly with the bases of DNA. Porine proteins that form holes in membranes are composed of beta sheets, arranged to form a pore in the membrane. And then finally, in hemoglobin, alpha and beta globin peptide chains make up the final molecule. Each have characteristic regions of secondary structure. The final folded shape of a globular protein is its tertiary structure. This tertiary structure contains regions that have secondary structure and determines how these are further arranged in space to produce the overall structure. A protein is initially driven into its tertiary structure by hydrophobic exclusion from water. Ionic bonds between oppositely charged R groups bring regions in close proximity, and disulfide bonds lock particular regions together. The final folding of a protein is determined by its primary structure, the chemical nature of its side groups. Quaternary structures. So when two or more polypeptide chains associate to form a functional protein, the individual chains are referred to as subunits of the protein. The arrangement of these subunits is termed quaternary structure. In proteins composed of subunits, the interfaces where the subunits touch one another are nonpolar, and they play a key role in transmitting information between the subunits about the individual subunit activities. Remember, the protein hemoglobin is composed of 2 alpha chain subunits and 2 beta chain subunits. Each alpha and beta globin chain has a primary structure consisting of a specific sequence of amino acids. This then assumes the characteristic secondary structure of alpha helices and beta sheets that are then arranged into specific tertiary structure for each alpha and beta globin subunit. Lastly, these subunits are then arranged into their final quaternary structure. This is the final structure of the protein. For proteins that consist of only a single peptide chain, the enzyme lysozyme, for example, the tertiary structure is the final structure of that protein. So now you have the levels of protein structure, and you see figure 3.22. So I won't get into motifs or domains, though what I will get into quickly are chaperones. So chaperone protein deficiencies can be implicated in certain diseases in which key proteins improperly folded. Cystic fibrosis is a hereditary disorder in which a mutation disables a vital protein that moves ions across cell membranes. As a result, people with cystic fibrosis have thicker than normal mucus. This results in breathing problems, lung disease, and digestive difficulties, among other things. One interesting feature of the molecular analysis of this disease has been the number of different mutations found in human populations. Many of these mutations affect the ability of the encoded protein to fold properly. The number of different mutations that can result in improperly folded proteins may be related to the fact that native proteins often fail to fold properly. So there you have it, the chaperone. Denaturation. If a protein's environment is altered, the protein may change its shape or even unfold completely. This process is called denaturation. Proteins can be denatured when the pH, the temperature, or ionic concentration of the surrounding solution changes. Denatured proteins are usually biologically inactive. This action is particularly significant in the case of enzymes, because practically every chemical reaction in a living organism is catalyzed by a specific enzyme. So this is vital to that cell's enzymes work properly. Traditional methods of food preservation, salt curing, and pickling involve denaturing of proteins. So denaturation of proteins is again involved in food preservation, salt curing, and pickling. Prior to the general availability of refrigerators and freezers, the only practical way was to keep microorganisms from growing in food was to keep the food in a solution containing a high concentration of salt or vinegar, which denatured the enzymes of most microorganisms and prevented them from growing on the food. Most enzymes, before I get there, let's go back to this part. So most enzymes function within a very narrow range of environmental conditions. Blood-borne enzymes that course through a human body at a pH of about 7.35 to 7.45 would rapidly become denatured in the highly acidic environment of the stomach. Conversely, the protein-degrading enzymes that function at a pH of 2 or less in the stomach would denature in the relatively basic blood. Similarly, organisms that live near oceanic hydrothermal vents have enzymes that work well at these extreme temperatures. They cannot survive in cooler waters because their enzymes do not function properly at lower temperatures. Any given organism usually has a tolerance range of pH, temperature, and salt concentration. Within that range, its enzymes maintain the proper shape to carry out their biological functions. When a protein's normal environment is reestablished after denaturation, a small protein may spontaneously refold into its natural shape, driven by the interactions between its nonpolar amino acids and water. This process is termed renaturation, and it was first established for the enzyme ribonuclease. The renaturation of ribonuclease led to the doctrine that primary structure determines tertiary structure. Larger proteins can rarely refold spontaneously, however, because of the complex nature of their final shape, so this simple idea needs to be qualified. And it's important to distinguish denaturation from dissociation. For proteins with the quaternary structure, the subunits may be dissociated, i.e. separated, without losing their individual tertiary structure. For example, 4 subunits of hemoglobin may dissociate into 4 individual molecules without denaturation of the folded globin proteins. They readily reassume their 4 subunit quaternary structure. So that has been proteins. And now let's end the chapter with our 4th and final macromolecule, the nucleic acids. The biochemical activity of a cell depends on the production of a large number of proteins, each with a specific sequence. The information necessary to produce the correct proteins is passed through generations of organisms, even though the proteins themselves are not inherited at all. Nucleic acids carry information inside cells, just as hard drives contain the information in a computer, or the GPS system in a car displays information needed by travelers, or just like the information that is saved on the storage devices within your cellular phone and computer. 2 main varieties of nucleic acids are deoxyribonucleic acid, known as DNA, shown in figure 3.13a, and then you have 3.13b. Then we have ribonucleic acid, or RNA. So you have a scanning tunneling micrograph of DNA at 3.13a, and then at B is this space filling model showing DNA at B. So both of those are DNA shown to you there. Genetic information is stored in DNA. I repeat, genetic information is stored in DNA. While RNA plays a variety of roles in the cell, including short-lived copies of genetic information used to direct synthesis of proteins. I repeat, RNA includes short-lived copies of genetic information used to direct the synthesis of proteins. So protein synthesis. Unique among macromolecules, nucleic acids are able to serve as templates for producing precise copies of themselves. This characteristic allows genetic information to be preserved during cell division and during the reproduction of organisms. Unlike DNA, which functions to store information, RNA has a variety of roles in cells. It can carry genetic information. It is an integral part of the machinery for protein synthesis. And it can also function to regulate the process of gene expression. This is RNA, you all. So as a carrier of information, the form of RNA called messenger RNA or mRNA consists of transcribed single-stranded copies of portions of DNA. These transcripts serve as blueprints specifying amino acid sequence of proteins. This process will be further described later on in chapter 15. Nucleic acids consists of long polymers of repeating subunits called nucleotides. Each nucleotide consists of 3 components: a 5-carbon sugar, a phosphate group, and an organic nitrogen-containing or nitrogenous base. In DNA, the sugar is deoxyribose. In RNA, it is ribose. Please see figure 3.14, and I'll go to that figure in just a moment. Nucleotides have 5 types of nitrogenous bases. 2 of these have a large double ring called purines that each are found in both DNA and RNA. The purines are adenine and guanine. The other 3 bases are single ring molecules called pyrimidines that include cytosine. So, in both DNA and RNA. Thymine in DNA only, and then uracil in RNA only. So again, the other bases that have a single ring are referred to as pyrimidines. They are cytosine, thymine, and uracil. Cytosine is in both DNA and RNA. Thymine is only in DNA, and uracil is only in RNA. Polynucleotides are formed by joining the phosphate of one nucleotide to a hydroxyl group on the sugar of another nucleotide in a dehydration reaction. We call this a phosphodiester bond because the 2 sugars are linked by ester bonds to a phosphate. A nucleic acid then is simply a 5-carbon chain of sugar linked together by 5 phosphodiester bonds with a nitrogenous base protruding from each sugar. And that is shown in figure 3.16. So this is figure 3.14, showing you those 3 components of the nucleotide. We have the phosphate group to the left, the 5-carbon sugar being ribose in RNA, being deoxyribose in DNA, and in that top middle, we have that nitrogenous base. So since there are 2 rings there, that would be a purine. So this is now figure 3.15 and 3.16. DNA stores genetic information. Organisms use the nucleotides in DNA to encode the information specifying the amino acid sequences of their proteins. This method of encoding information is very similar to the way in which sequences of letters encode information in a sentence. A sentence written in English consists of a combination of the 26 different letters of the alphabet in a certain order. The code of DNA consists of different combinations of the 4 types of nucleotides in specific sequences, such as C, G, C, T, T, A, G, C. DNA molecules in organisms exist as 2 polynucleotide chains wrapped about each other in a long linear molecule in eukaryotes and in a circular molecule in most prokaryotes. The two strands of DNA polymer wind around each other like the outside and inside rails of a spiral staircase. Such a spiral shape is called a helix, and a helix composed of 2 chains is called a double helix. Each step of DNA's helical staircase is composed of a base pair. The pair consists of a base in one chain attracted by hydrogen bonds to a base opposite it on the other chain, and this is shown in figure 3.17. The base pairing rules arise from the most stable hydrogen bonding configurations between the bases. Adenine pairs with thymine in DNA, and adenine pairs with uracil in RNA. And then cytosine pairs with guanine. That is complementary base pairing. So the bases that participate in base pairing are said to be complementary to each other, hence complementary base pairing. Additional details of the structure of DNA and how it interacts with RNA in the production of proteins will be taught to you on the 14th and 15th chapters. This is now figure 3.17, showing you the double helix of DNA, the structure of DNA, along with base pairing, with those 3 hydrogen bonds between cytosine and guanine, and those 2 hydrogen bonds between thymine and adenine. RNA is similar to DNA, but with 2 major chemical differences. And this chapter is ending here really, and I repeat, really soon. First, RNA molecules contain ribose sugars, in that the C2 is bonded to a hydroxyl group. Second, RNA molecules use uracil to replace thymine, or in place of thymine. Uracil has a similar structure to thymine, except that one of its carbons lacks a methyl group. RNA is produced by transcription from DNA and is usually single-stranded. The role of RNA in cells is quite varied. It carries information in the form of mRNA. It is part of the ribosome in the form of ribosomal RNA, or rRNA. It carries amino acids in the form of transfer RNA, tRNA, and then there has been a revolution of late in how we view RNA, since it has been found to function as an enzyme, and other forms of RNA are involved in regulating gene expression. So other nucleotides, and excuse me, this is showing a comparison of DNA structure to RNA structure. At the top is DNA. It's a double helix. Adenine, guanine, thymine, cytosine. Well, at the bottom, it is showing you RNA. Uracil, adenine, cytosine, guanine, adenosine, uracil. So single-stranded RNA, double-stranded, double helix DNA. Nucleotides play other crucial roles in cells. For example, adenosine triphosphate, known as ATP, shown in just a moment here at figure 319, there it is at the bottom, is called the energy currency of the cell. Cells that use the energy released by breaking down food molecules to synthesize ATP. Cells can then use the energy released by ATP hydrolysis in a variety of transactions as we use money in society. Energy from ATP hydrolysis is used to drive energetically unfavorable chemical reactions to power transport across membranes and to power the movement of cells. 2 other important nucleotide-containing molecules are NAD+, or nicotinamide adenine dinucleotide, and FAD, or flavin adenine dinucleotide. These molecules function as electron carriers in a variety of cellular processes. You get to those later on with respiration and photosynthesis. So this has been your instructor, Skylar Huff,